My name is Kai. My partner is William. Today we're going to present CYK4, a subunit of a larger complex that inhibits Rac1. And through Rac1 inhibition, it also influences PAK1 and ARHGEF7 effector pathways during cytokinesis. So there's a lot of adhesion going on during cytokinesis, and they're actually competing processes. Cells lose adhesion to surrounding surfaces, losing their leaf-like structure and adopting a sphere-like conformation. And then cells also gain adhesions at the cell equator, forming the cleavage furrow and the contractile ring. Rho A and those family of GTBases are central to cytokinesis. They recruit effector proteins, they promote actin polymerization and contractility, they form the cleavage furrow, the physical characteristics of the little furrow at the center of the cell before it divides, and it's localized in the equatorial region as well. ECT2 is a Rho A GEF. We know that GEFs are guanidine exchanging promoting factors. So the Rho A GTPase, when interacting with EC2, will bind to a GTP molecule and be activated. Once activated, it'll recruit the microtubule-bound central spinolin complex, or it'll interact with the microtubule-bound central spinolin complex and phosphatidyl phosphates and the inner leaflet of the plasma membrane. It effectively localizes Rho A to the midpoint of dividing cells. Because there is this competing process between gain of adhesion and loss of adhesion, there's suspected activity of gaps involved in this process as well. So GEFs will increase the probability of a cell to form these adhesions, but because we see basically that cells also lose adhesions, that there is a gap that plays a role as well. A candidate gap, and it's been strongly implicated in causing cells to lose these focal adhesions, is the CYK4 subunit of the central spinolin complex. It contains a gap domain within its primary, primary coding sequence. There are several problems though with its candidacy. Biochemical analysis of CYK4 subunit has found that it has little activity towards Rho A and greater activity towards Rac1. That's implicated by genetic evidence in D-melanogaster and gap activity. Furthermore, gap activity of CYK4 can be dispersed under some conditions. CYK4 gap mutants grow and divide without obvious defects. Mutations that reduce gap activity result in cytokinesis defects that can be suppressed by depletion of Rac. So CYK4 may directly limit Rac rather than Rho A activity during anaphase, and this is one of the competing hypotheses with it being a gap for Rho A. To test the alternative hypothesis that it's a gap for Rac1 rather than Rho A, they hypothesize CYK4 is a crucial regulator of Rac1 in adherent cells, and that inhibition of Rac1 dependent effector pathways in anaphase is needed for cytokinesis.
All right, so that kind of takes us into what this paper actually investigates. So like we did in class a lot of times, we like to look ahead at like the ending model figure that they kind of sum things up with to understand things better. So what we're looking at is this is what was previously known with like Rho A being activated with the GEF to form this furrow, and with the contractile forces that we learned about in the first couple papers. And CYK4, the paper, the protein, the gap we're looking at in this paper is important for inhibiting Rac1 pathways that are responsible for the adhesion formations and spreading of the cell that happens when the cell's usually not dividing. But in mitosis, there's this competing event of where there needs to be a furrow formation, and where adhesions are being lost on the cell surface but being formed inside the contractile ring. So we're focusing in on that furrow and how we're inhibiting that spreading. So the paper had a lot of figures, and we just wanted to grab like important ones from the text, and if you guys have any questions, that's fine. We can dive more in depth.
Yeah, going off William said, there are a lot of other figures that investigate different uh, uh, motifs of Rac1 dependent regulation. They looked at serine residues for regulating Rac1 itself. They looked at how whether what kind of downstream effectors it also affects. We particularly looked at we specifically looked at Rac1 and its how it's influenced by CYK1 and ignored its how it's regulated and other probably important facts about this regulatory mechanism. Anyways, figure one just looks at CYK1's gap effect on several GTPases. It looks so if you see on the bottom left and C, RAC1 and CDC2 are affected by CYK1. You can see changes in their GTPase activity according to the y-axis, which looks at GTP hydrolysis. You can see a dip in metaphase and for RAC1, which is expected. Row A and RAB1 are unaffected by CYK1, and you can see the same thing for figure D. Both of these just corroborate the fact CYK1 is a gap that affects RAC1 and CDC42.
And so looking at figure 3, this is where they started to actually look at images and image J using staining. So we learned about like binucleated cells as a good readout for failed cytokinesis. So in A, we were looking at the effect of CYK4 and how its importance in proper cytokinesis. So you can look at these images and you can see these binucleated cells that aren't properly dividing. And
Alright,
so that this is just a bar graph of that readout.
I have a quick question.
Yeah,
Why are some of those nuclei like pink, purpley, and some blue?
I think that's just part of the staining because like they had the blue, I'm assuming is the DAPI staining, and then they have I guess like a reddish pink, and I'm not really sure why there's a mix. It just could be because they're overlapping, perhaps.
Yeah, I'm just it's so the induced what like it because they only show up in the induced. I'm just curious what that is. It's
There's yeah, I
I'm
think
not,
that's just the activities being of the CYK4.
so that's actually CYK4 that we're
Yeah, I
looking
think
at.
these are controls. So there's
Okay.
not, we're looking mostly at these.
Okay. Yeah. I get that. Okay. Cool. We can move on.
And then in B, it's more staining, but this time we're focusing on the Rac1 activity. And you see where there's more binucleated cells is where there's uh, so of the wild type and more of this mutant Rac1. So when Rac1 isn't properly interacting with CYK4 and it's not properly regulated, you get this failed cytokinesis. And then figure 7 was
Sorry,
using.
I had a question.
Yeah.
I had a question. So the Q to L mutation, that's an overactivation of RAC1. That's like constitutively active RAC, always on RAC, or always on RAC.
It's either, I think it's inhibiting the way that it can be. Yeah, it can be regulated by CYK4, so it can't be
Turned off
because it needs to be inhibited at the cleavage furrow.
Yeah.
For
That's it.
proper
So
cytokinesis.
So that is RAC expression always on, and then like look at those cells, like look how big and spread out they are.
Yeah.
You know, because RAC helps spread cells out through migration and stuff. So that's interesting. Cool. I'll shut up again.
And then figure 7, we're just looking at electron microscopy. And it's like looking at just an easy way to see over time how like the cleavage furrow is like proper, and like when you have a wild type CYK4 to regulate that Rac1 activity. But then when you have like this mutant CYK4, when the gap activity can't properly function, you get this weird, like improper furrow formation, and large like cells that haven't blocked this spreading. And there was another figure right after this that is like very similar images. It just focuses in on the Rac activity. So kind of like what the last figure showed, like there's more Rac activity, which is leading to this large cells that are failing cytokinesis.
There's like, if you look at that 60-minute mark, there's such a huge difference in the way those cells look, right?
Yeah.
And that little thing between them, I'm not sure if we've not really talked about that, but that little tiny like spindle between them is basically a bunch of microtubules and leftover actin and a bunch of other proteins that really do the pinching off part. It's called abscission. It's like this little like membrane bridge that's stuffed full of microtubules. And there's a bunch of sick CYK4 in there, which is doing all kinds of things. So cool. Great. Keep going. Sorry. I just, you said that cell difference and it just really stands out, the cells, the difference. And then they never cut that little bridge. So they just become that two cells. So it's like the cells are able to cytokinese almost all the way. They're just not able to cut that final little step. It's pretty cool.
Right.
Which MDC is super important for helping maintain that structure, that small little filamenty structure. So it makes sense that if we mess with that balance, it's going to mess it all up. Cool. Okay. Now I will really shut up.
And then figure 9, we're looking at more so the adhesions that are actually supposed to be either forming or breaking during mitosis. So normally you would expect the adhesions on the cell surface that are normally there to like kind of be localized more so at the central like equator in a cell furrow, because that's where all like the activity kind of needs to be happening, and you want to be forming the sphere shape on the outside. You don't want to be like in that natural like just like adhesion to like all the other cells around you or outside the cell surface. And so what we see is in the mutant with CYK4 that there's still we use vinculin as a stain for this. So we learned about the vinculin's importance in the adhesion mechanisms. And so we can see that there's a lot of localization on the cell surface still. So there's just not proper regulation of those adhesions that are supposed to be broken so that proper mitosis and cytokinesis can occur.
Yeah, like so now we have all these adhesions that are being maintained to the floor, but the cell's trying to divide. That's
right.
Really, yeah.
Because like the whole point of this paper was looking at how this, during mitosis and especially anaphase and cytokinesis, you're supposed to be forming like this, like you're forming and breaking adhesions at the same time. You're contracting and spreading at the same time, like how these like gefts and gaps regulate. And so like this was focusing in on like CYK4 as an important gap in this overall mechanism.
Yeah, and like its power to like organize the like activity map of the gap is so important. That's cool.
And so that kind of just ties into what I kind of just said it with the conclusion and the impact of the results is we know we've learned about Rho A and its importance in the contractility and forming this furrow. We've talked about today about like Rac1, like this, the cells spreading, its importance, and how there's gaps and gefts that are important for this activating and inactivating that need to happen simultaneously, like during mitosis, because there's a lot going on when a cell's trying to divide. It's trying to like change its shape. It's trying to like form two cells, and it's trying to break and form adhesions. So this is just like drawing another step in that overall mechanism.
And this paper was also published in 2012, so I assume we've come a long way since then, finding other gaps responsible for promoting adhesion loss during cytokinesis.
And then our model is kind of like, it kind of ties into our discussion activity where we're going to ask you guys to like kind of at least think about, and if you can like draw out like what you guys see with like what's happening with the cell, where the adhesions are forming and breaking. So like ours was just kind of like we have like this Rho A is like contractile, like pushing in on the ring to form that furrow that we've learned about so many times. And then Rac1 in red is pushing out the cell, making it so it can spread and form these adhesions, which is in orange, but I guess it's kind of hard to see between the red. And then like CYK4 is like inhibiting Rac in the furrow because you want proper Rho A activation and contractile forces in the furrow. You don't want to be spreading and forming adhesions on the cell surface.
It's such a nice little like geography lesson in like activity of GTPases and how they're affected by this one protein and how it results in these huge dramatic differences in what's happening in those cell parts.
Yeah, and so our discussion activity was just like if you guys can just to like take a moment to think about and draw up for yourself, because I think that's the best way to understand is to be able to come up with it yourself, like the adhesions that are forming and breaking.
Do we want to use that online whiteboard thing?
Yeah, I like we tried to figure out how to work that, but I couldn't. I didn't even remember it.
Okay. I can remember it. I mean, it's like almost like WhatsApp. It's like it's like app, yeah. I had to remember it. Okay, let me start drawing.
I also thought like if that might take too long, like just to, like if anyone has like a paper, we could do that. Get breakout room for a second, just like.
Yeah, I've got it right now, so we should be good.
Oh, okay.
Okay, so that should be the link, and then we can all just start drawing in there. Let me know if it doesn't work. Okay, so you want us to draw, what are the two things? Just with and without cyc?
So like the cells when it's during I don't know why I can't do during like anaphase and like cytokinesis, like these adhesions are having to break, and like what you guys would like maybe you can look at what they saw more like when we talked about with this, like what exactly is happening when like you see these large cells that can't quite properly like break. If that makes sense.
Yep, that makes sense. Okay, let's see.
I'll just leave this figure up because I think it's the.
That's good.
I guess. Right.
We need some like cell drawing music.
Mm-hmm. We were having trouble playing audio before you joined, so I don't know if that would go well.
I'm like trying to draw a bunch of dots, and this thing doesn't want to like keep any of the dots. It's funny. It keeps deleting them.
And that's all we had. So
Yeah, I mean, you guys, whenever you're ready, you can. It's up to you. You guys are in charge. You can bring us back.
I think we can come back if you guys want.
Yeah.
Does anyone have any questions other than?
Yeah, any questions?
Did they look at abscission? Oh, sorry, I'm not. Did they look? Let me see. Oh, I am unmuted. Did they look? Yeah, did they look at the type of failure, or kind of like to get at what's causing the failure? Like, do the cells just like
I think
regress?
that was with the. I think that's what we saw with like the large cells and such. Like with the, like you could see it took them longer to kind of like for that furrow to like kind of form, I guess. And I think it kind of made sense to me that like if you weren't inhibiting the thing that's like trying to maintain like cell adhesions and like go against contraction, then like that it would take longer to actually like contract and like
Yeah,
it would like lead to a larger cell, I guess. Sorry.
I don't know. I mean, they probably have since then, but in this paper particularly, they couldn't pinpoint the exact mechanism behind or the exact mechanism at which point the sort of loss of function actually occurred, leading up to the binucleated phenotype. So we know that they used the binucleated phenotype as a readout, but behind that, up to like to what point behind that link, like at what point within that chain of causation, Rac1 distorting Rac1 actually affected the final phenotype, because Rac1 affects multiple downstream effectors. So
Yeah, like they have a lot. Sorry, keep going.
no, that was all I really had to say. Is that we know what Rac1 affects, but we don't know how much that contributes to the final phenotype. We just know that at some point it does impinge on proper precision.
Yeah, like can someone show that figure again? That like montage of the dividing cell.
Do you guys see the presentation, or you just see the?
I see it.
Okay.
I need to full screen this. Okay. Yeah. So like, yeah, that's a good point. So like, we have these different phenotypes. Like those cells are spreading out more, but they're also never cutting that bridge. And so I guess I'm curious, like, is the spreading out causing them to not be able to cut the bridge, or are those separate? You know, are those interplaying with each other, or are they distinctly both messed up, but both separate from each other, messing up each other?
I think that's like kind of thing the paper was looking at was like there's row A and Rac1, both are like there's GEFs and gaps for both.
Yeah.
And so like Rac1 is the cell spreading and adhesion that's being inhibited here. So
but I mean, I just know from like personal knowing of like CYK4 and stuff. Like if you mutate its like ability to bind to the membrane, for example, it'll also cause failure similar to this. Like they're never able to cut that little bridge, and that's why they fail. But that could also be affecting cell spreading too. So and there's other cells called like slime molds, like dictyostelium, and they don't even have myosin 2 at the cleavage furrow, and they have it, but you can get rid of it, and they can still divide because they're like crawling away from each other so much that it breaks them apart. So like that question of like what is actually the forces that are breaking the two cells apart? Is it actually like the crawling away, or is it actually like forces generated from the machinery that accumulates in that little bridge? I think those are related for sure. And we're still, I mean, this paper came out in 2012. We know more about it since then, but not a ton more. So I was just curious kind of what your thoughts on that were, if they talked about that at all.
I think one of the figures we showed, where they stained for a abnormal distribution of vinculin. Yeah, this one right here.
Yeah.
We can start testing either further downstream or upstream of a mutant CYK4 subunit. So then by manipulating either further downstream, and then at one point, once manipulation no longer affects abscission, we can use that to determine, okay, this is where sort of the extent of CYK4 influences cytokinesis. You know what I mean?
Yeah, totally. So you're saying like
yeah,
like a more thorough like watching of the cells dividing over time.
Right, yeah, because all we can really do is catalog the effects of every individual manipulation we do. We can't really construct from scratch the entire mechanism.
Yeah, we'd have to like recreate it.
Yeah,
Piece by piece.
Yeah, exactly.
One interesting thing I think is like, could we disrupt these? Could we disrupt these focal adhesions a little bit? And would that maybe rescue the sick mutant or the sick depletion? Does that make sense?
Yeah,
like
I think that would be an interesting experiment. Yeah, because they most proximately, so Rac1 is right next to right downstream from CYK1, and so Rac1 rescue mutants have been observed, but we can also go further downstream to see, well, what does Rac1 affect? Maybe rather than compensating for Rac1 deficiencies, we go downstream and compensate for those deficiencies that Rac1 is originally responsible for.
Right. Right. Because we'd be like potentially, well, not inhibiting Rac, but if overactivating Rac by knocking down the gap. It's like, this is like another, it just really shows you the crazy balance that is happening in all these cells, because this balance is also happening in conjunction with like the anilin P190 row balance.
Right, yeah.
And those are just two of the GTPases involved.
Right, and they probably intersect too by some.
They definitely do, because I mean, CYK4 on its own binds to PAK2 as well, right? Which is the gap that's turning on, or the GEF that's turning on row A. So it's this interconnected web of gap activity for sure. Yeah, I guess it would be cool. I kind of want to know a little bit more about those just personally. So I think it would be cool if they did an experiment where they like messed with the focal adhesions and see if they could rescue some of what they saw. Okay, let's talk about the drawing. Sorry, I got all.
No, all good.
These are interesting.
Yeah, we just kind of wanted people to think about it for themselves a little bit.
Yeah. What was the person thinking? This blue and red one stands out to me with this stuff outside of the cell. I'm just curious what.
That might be like adhesions being broken.
Yeah, I guess that could be the adhesions between the cells.
Yeah, I agree with that. It's the adhesions being broken, but a little bit of the cell's still connected and can't get break off.
I see.
Oh yeah, that's adhesion.
Anybody else have stuff that they wanted to talk about with these? So with mine's the like toppy right one ish. There's like, has that little C and then sick. Yeah, that one. So the way I was thinking about it is like just showing that change in focal adhesions, and like the cells being a little more adherent in the control versus the disruption. And the little purple things represent the focal adhesions. Yeah.
Jeez.
Cool. Okay. Did we have anything else? Okay.